Here in Chapter 7, we've been discussing Mendelian and quantitative traits. Thus far in the chapter, the following topics have been addressed: Mendelian genetics, the inheritance of traits, and how we have our genetic diversity. I did not do a video lecture on this, but it was covered in your book, both quantitative genetics as well as how the genes and the environment interact along with the individual for that individual to have a unique genetic profile. One thing not discussed in the book is how we can modify the epigenome, which can lead to altered gene expression. And this is one additional way that we have our unique genetic profiles. So I wanted to briefly cover that topic since I think it's so interesting and it is not addressed at all in your text. What is epigenetics? Well, it's the examination of changes in gene expression that are not caused by changes in the DNA sequence itself. So when I say DNA sequence, I'm talking about the A's and T's and C's and G's. We are not changing the nucleotides, but we are causing changes to the DNA or the histones around which DNA is wound up to form our chromosomes. So the DNA can have the addition of a methyl group. This process is called methylation. So you still have the A's and T's and C's and G's, but you simply attach onto those nucleotides a methyl group, which is a CH3 group. Histones can be modified as well. They can also be methylated or acetylated or phosphorylated, and I'll show you what those different groups look like. So here is showing you how the DNA itself can be modified or the histones can be modified. So at the top of this picture, you can see a methyl group, which is just a CH3 group, has been attached to this cytosine that's comprising this DNA ladder that you see. At the bottom of the figure, you can see that we ultimately can form our chromosomal structures by the way that we wind the DNA around different types of proteins, predominantly histones. These histones can be modified as well. What do these actual modifications look like? Well, a methyl group is the addition of a carbon with 3 hydrogens. You can also have the histones be acetylated, which is the addition of this side chain, or phosphorylated, which is just a phosphorus and 4 oxygens. So these modifications completely change the ability of that gene to be turned on or turned off. So it can alter gene expression without actually changing the sequence of the nucleotides in a particular gene. Here's showing you one more picture of those types of modifications. In figure A, what you can see is the chromatin is getting modified. You can either add the methyl groups, which is indicated here with the red circles, or it can be demethylated, which is the white circles. So the histones can be methylated as well as the DNA itself. In this bottom image, what you can see is this gene is turned on or turned off depending upon whether or not it is modified in a particular way. So in the top image, this is showing that the gene is actually turned on. You have access to the gene. This is when you see that the histones are acetylated and the histones are also unmethylated or demethylated. In the bottom part of this picture, the gene is turned off. You can see that the histones are much more condensed, and you actually just don't have access to the DNA. So we have this condensed structure, and in this image, the histones are highly methylated. So now that you have a very brief introduction to epigenetics, I want you to now go and look at the 2 videos to which I've created a link. The first is on twins and their studies of how their epigenomes begin to diverge and differ as they age. So you can see for identical twins, at birth their genomes and their epigenomes are identical. However, as they age, their genomes might stay the same, but their epigenomes differ, which can lead to different disease states. After completion of that video, you to then look at an interactive, which I've also created a link for that as well, looking at some animal studies where they try to study the epigenome. You'll also find attached at the bottom of this document an assignment sheet for you to fill out, and you can then submit that directly onto Canvas. I hope you enjoy these videos. All the best.